Welcome to all. Today we discuss about RDNA technology. Recombinant DNA technology is a technique that alters the phenotype of an entity when a genetically modified vector is introduced into host incorporated into the genome of the host. Thus the process entails introducing a foreign fragment of DNA into the cell's genome containing the desired gene. This particular gene that is introduced is referred to as the recombinant gene, and the technique is known as recombinant DNA technology. Recombinant DNA technology involves the selection of the desired gene for administration into the host, followed by a selection of the perfect vector with which the gene has to be integrated, and hence the recombinant DNA is formed. This recombinant DNA then has to be introduced into the host, and at last it has to be maintained in the host and carried forward to the offspring. Plasmid. A plasmid is a small circular piece of DNA that is different than the chromosomal DNA, which is all the genetic material found in an organism's chromosomes. It replicates independently of chromosomal DNA. Plasmids are mainly found in bacteria, but they can also be found in archaea and multicellular organisms. Plasmids usually carry at least one gene. Many of the genes that plasmids carry are beneficial to the host organisms. Cloning vector. A vector that's related to molecular biology is a DNA molecule that is used as a vehicle to carry a particular DNA segment into a host cells. We may call DNA vehicle carrier. Types of vector: BBR322, cosmids, shuttle vectors. BBR322, the plasmids, was one of the first EK2 multi-purpose cloning vectors to be designed and constructed for the efficient cloning and selection of recombinant molecules in Escherichia coli. Cosmids. Cosmids can be used to clone DNA fragments up to 45 kb in length. A cosmid is a type of hybrid plasmids that contain a lambda page cos sequence. Shuttle vectors. A shuttle vector is a vector constructed so that it can propagate in two different host species. Therefore, DNA inserted into the shuttle vector can be tested or manipulated in two different cell types. Next one is the PUC19. The molecule is a double-stranded circular DNA. It has 2686 bases in length. The PUC19 plasmid confers ampicillin resistance and completed defects in beta-galactoses in appropriate host strains. Next one is the bacterial base, bacterial chromosome, yeast artificial chromosome. Molecular steps: isolation of insert and target DNA fragments, and ligation of the inserts into suitable vector plasmids, and transformation of recombinant plasmids into the host for multiplication. Identify the correct host cells carrying the recombinant molecules. We have to choose the host organism and cloning vector, and preparation of vector DNA, preparation of DNA to be cloned, creation of recombinant DNA with DNA ligase, introduction of recombinant DNA into host organism, then selection of organisms containing vector sequences, selection and screening for recombinants. After the introduction of recombinant DNA into the suitable host cells, it is essential to identify those cells who have received the DNA molecule. This process is called screening. The vector or foreign DNA present in recombinant cells expresses the characters, while the non-recombinants do not express the character or traits. For this, some of the methods are used, and one such method is blue-white selection method. And next one is the insertion inactivations, antibiotic-resistant markers, replica plating techniques, molecular techniques, electrophoresis. Electrophoresis is a separation technique used to separate different biomolecules with positive and negative charges. Nucleic acid hybridization. We have blackening techniques. These are all techniques used for selected and screening of recombinants. Next one is the application of RDNA technology. Transgenic plants are the best example of RDNA. For example of pest resistance, virus resistance, herbicide tolerance, and stress tolerance. Example: cold, heat, and salt. Delayed fruit ripening and genetically programmed. We are physiologically biochemical changes. We are doing the fruit ripening. Molecular mechanisms controlling the ripening of fruit. Transgenic animals. The production of transgenic livestock has the opportunity to significantly improve the human health and enhance nutrition, protect the environment, increase animal welfare, and decrease the livestock diseases. Thank you.